So you should have looked at the process of transcription, and now the following process, the final stage of protein translation, is translation. So once a molecule of the mRNA has been transcribed, as you mentioned in the previous video, it moves out the nucleus via a nuclear pore. In the cytoplasm, the mRNA combines with the ribosome. That is the cellular structure which will make a polypeptide chain. So a ribosome is made of two subunits. It's made of rRNA, which is ribosomal RNA, and it's made of ribosomal protein. It's about 65% of it is the ribosomal RNA. Now this process that takes place is called translation, and the ribosome essentially is going to link together multiple amino acids that are carried in by the tRNA molecules based on the mRNA strands codons. So as we said, the mRNA attaches itself to a ribosome, and tRNA molecules will now carry amino acids to it. As we mentioned in one of the previous videos, ATP provides energy to attach an amino acid to what would be called an empty tRNA molecule. So it's important that energy is involved in this process, preparing these amino acids and tRNA molecules. So this will bind to the correct place on the anticodon, onto the codon. It has a complementary anticodon on the tRNA, which will exactly match the codon. If you recall before, I said that it's quite obvious when you look at this that you can't have an overlapping code. Well, this is why. Because one amino acid is going to fit to its first codon, and the ribosome will then put a second tRNA into it as well. They can't overlap because they're both present at the same time. So the second tRNA molecule attaches itself to the next codon on the mRNA in exactly the same way as what we've seen before, and a peptide bond forms between the amino acids. The empty tRNA molecule will now dissociate, will now move away from the ribosome, and it will leave a space here to, if we're looking at it from our perspective, to the left. The whole ribosome will now move along to the right, so that will cause this tRNA staying in its same place to be in the position that the empty one has now moved out of. The amino acid will start to move away from the ribosome, and we'll then create an empty space here to the right. That can then be filled with another amino acid, and the process continues over and over as the ribosome moves along. So the one on the left will go away once the peptide bond's formed, and the ribosome will move along. Another one will enter into the next codon, and another peptide bond will form between the amino acid, and the process will continue until you make a nice long polypeptide chain. That continues until, as you'd have seen in one of the other videos, if you're not sure you can always look back, it will happen until there is a stop signal met on the mRNA molecule. There is no tRNA for that, so that means that the sequence will finish and the ribosome will move away and the polypeptide chain will be complete. On this process here, I'll give you a moment to kind of write down and number these into the correct order, and then I will show you the correct order so that you can check if you got it right. This is obviously a very generic, well, not a generic, a very simplified version of the process, but it might help you get started with revision. Before we do that though, it's probably worth pointing out, while I've just remembered, that this process, there can be multiple ribosomes moving along the same RNA sequence, so that can greatly speed up the process. Okay, back to the question. So I will leave you now. I'll give you a few seconds to pause it, and then I'll give you a little bit of a warning, and we'll see the answers. Okay, so here are the answers for you to check yours, and that brings us to the end of the translation video and also the end of the genetic code in terms of transcription and translation.